Hello and welcome to Easy Learning with Dr. Salman Khan. In this video, we will be studying the parasite Trichomonas vaginalis under the following sections: Introduction, Epidemiology, Habitat, Morphology, Life Cycle, Modes of Infection, Pathogenesis, Clinical Features, Laboratory Diagnosis, and Prophylaxis. First, the introduction to the parasite. Trichomonas differs from other flagellates as they exist only in trophozoite stage. Cystic stage is not seen. Genus Trichomonas has 3 species which occur in humans. Those are: first, Trichomonas vaginalis, second, Trichomonas hominis, third, Trichomonas tenax. Then the history. Trichomonas vaginalis was first observed by Donne in the year 1836 in vaginal secretion. Then epidemiology. The prevalence of trichomoniasis varies from 5% patients at hospitals to 75% in sexual workers. Next, the habitat. In females, it lives in the vagina and cervix and may also be found in Bartholin's glands, urethra, and urinary bladder. In males, it occurs mainly in the anterior urethra but may also be found in the prostate and prepucial sac. Then the morphology. It exists only in the trophozoite stage. The trophozoite is pear-shaped or ovoid and measures 10 to 13 micrometers in length and 5 to 10 micrometers in breadth, with a short undulating membrane reaching up to the middle of the body. It has 4 anterior flagella and the 5th running along the outer margin of the undulating membrane, which is supported at its base by a flexible rod called the costa. A prominent exostyle runs throughout the length of the body and projects posteriorly like a tail. The cytoplasm shows prominent siderophilic granules, which are most numerous alongside the exostyle and costa. The trophozoite is motile with a rapid jerky or twitching type of movement. This is the trophozoite along with the structures it possesses. In total, 5 flagella along with the nucleus containing the nucleolus, an exostyle, siderophilic granules, costa, and the undulating membrane. This is the trophozoite of Trichomonas vaginalis when viewed under a microscope. Next, the life cycle of the parasite. The life cycle of Trichomonas vaginalis is completed in a single host, either male or a female. The trophozoite cannot survive outside, so infection has to be transmitted directly from person to person. Sexual transmission is the usual mode of infection. Trichomoniasis often coexists with other sexually transmitted diseases like candidiasis, gonorrhea, syphilis, or human immunodeficiency virus, also called the HIV virus. Vaginal pH of more than 4.5 facilitates the infection. The trophozoites divide by binary fission. As cysts are not formed, the trophozoite itself is the infective form. Next, modes of infection. The trophozoite cannot survive outside, so infection has to be transmitted directly from person to person. Fomites such as towels have been implicated in the transmission. Sexual transmission is the usual mode of infection. Babies may get infected during birth. Then the pathogenesis. The incubation period is roughly 10 days. Trichomonas vaginalis particularly infects squamous epithelium and not the columnar epithelium. It is an obligate parasite and cannot live without close association with the vaginal, urethral, or prostatic tissues. It secretes cystine proteases, adhesins, lactic acid, and acetic acid, which disrupt the glycogen levels and lower the pH of the vaginal fluid. The parasite causes petechial hemorrhage and mucosal capillary dilatation, characteristically called the strawberry mucosa. Also, metaplastic changes and desquamation of the vaginal epithelium. Intracellular edema and so-called chicken-like epithelium is the characteristic feature of trichomoniasis. Next is the clinical features. The clinical features are observed in males, females, and infants who are infected during birth. The incubation period of trichomoniasis is 4 days to 4 weeks. First, clinical features in males. Infection is often asymptomatic, particularly in males, although some may develop urethritis, epididymitis, and prostatitis. In females, cervical erosion is common. It may produce severe pruritic vaginitis with an offensive yellowish green, often frothy discharge, dysuria, and dyspareunia. Endometritis and pyosalpingitis are infrequent complications. In infants, rarely neonatal pneumonia and conjunctivitis have been reported in infants born to infected mothers. These are the clinical features. Next, the laboratory diagnosis. The laboratory diagnosis is done in the following ways. First, the microscopic examination. Second, direct fluorescent antibody. Third, culture. Fourth, serology. And fifth, the molecular method. First, microscopic examination. It is done in wet mount or permanent stain. Wet mount, vaginal or urethral discharge is examined microscopically in saline wet mount preparation for characteristic jerky and twitching motility and shape. In males, trophozoites may be found in urine or prostatic secretions, and abundance of leukocytes is seen. In permanent stain, fixed smears may be stained with acridine orange, Vapanicolau and Giemsa stains. Then direct fluorescent antibody or DFA is another method of detection of the parasite and is more sensitive than the wet mount. Next, culture. Culture is recommended when direct microscopy is negative and is considered as a gold standard as well as the most sensitive. It is 95% method for the diagnosis of Trichomonas vaginalis infection. It grows best at 35 to 37 degrees Celsius under anaerobic conditions. The optimal pH for growth is 5.5 to 6. It can be grown in a variety of solid or liquid media, tissue culture and eggs. Cysteine peptone liver maltose, that is CPLM medium, and plastic envelope medium PEM are often used. Then the serology. Enzyme-linked immunosorbent assay, that is ELISA, is used for demonstration of Trichomonas vaginalis antigen in vaginal smear using a monoclonal antibody for 65 kDa surface polypeptide of Trichomonas vaginalis. Then rapid immunochromatographic tests, ICTs, are now available for detection of antigen like OSOM Trichomonas rapid test, Xenostrip TB. And lastly, the molecular method. Deoxyribonucleic acid hybridization and PCR are also highly sensitive, that is 97%, and specific 98% tests for the diagnosis of trichomoniasis. Sensitive and specific commercially available nucleic acid amplification test, NAAT, also known as Aptima Trichomonas vaginalis assay, has been developed. Lastly, prophylaxis. Prevention is same as for other sexually transmitted diseases. Avoidance of sexual contact with infected partners and use of barrier method during intercourse prevent the disease. The patient's sexual partner should be tested for Trichomonas vaginalis when necessary. This is all about the parasite Trichomonas vaginalis. If you wish to see more of such videos, do comment below. And don't forget to like, share and subscribe to my channel. Also, press the bell icon for early notification of my new videos.